Hello students, in today's topic we will discuss photosynthesis in various plants which are termed as C3, C4 and CAM plants. The process of photosynthesis is the production of glucose molecule through water molecule, carbon dioxide, and in the presence of sunlight, plant produce their food. But this process is slightly differ in the C4 and CAM plants which are grown in the high temperature environment. So we have categorized the plants into three categories: C3, C4 and CM plants. C3 plants are wheat, soybeans, potato and rice, and about 70% of plants falls under this category. They have a typical anatomy. The process of photosynthesis is very basic in the C3 plants. The process of photosynthesis is divided into light reaction and dark reaction, and this dark reaction contains the Calvin cycle. When we talk about the C4 plants, they are grown in slightly higher temperature environment. They Some examples are maize, sorghum, sugarcane and millets. These C4 plants have adapted themselves to maintain the photosynthesis in the high temperature as well. So they have to avoid the photorespiration. C4 plants have Kranz anatomy which allows them to avoid the process of photorespiration and emphasize on the photosynthesis. CAM plants are grown in very high temperature areas such as deserts. They have mainly cactus family and pineapples. They grow in very high temperature regions, therefore they have to focus on the prevention of water loss, as well as they have to avoid the process of photorespiration. So they have a xeromorphic anatomy which allows them to prevent the water loss as well as the avoidance of photorespiration. We will see the anatomy and the process of photosynthesis in each category. Calvin cycle in C3 plants is a basic process of photosynthesis in which molecules of carbon dioxide are being fixed by the rubisco carboxylase. This rubisco converted into 3-phosphoglyceraldehyde, which is a 3 carbon molecule. That's why it is called as C3 plants. This Calvin cycle use the ATPs and produce the glucose molecule which act as a food for plants. This is the basic Calvin cycle. But in higher temperature condition, the rubisco enzyme takes the oxygen molecule instead of carbon dioxide, and this process is known as the photorespiration, which consumes the energy in the form of ATP. This is the unwanted process of plants that plant will avoid to save the energy. So the C4 and CAM plants have adapted themselves to avoid the photorespiration and emphasize on the photosynthesis. This is because of their special anatomy. When we see the anatomy of C3 plant, there are stomatas from which the air enters into the through the mesophyll cells, and which are loosely bound to each other, and there are vascular bundle surrounded with the bundle sheath cells. You can see mesophyll cells are very loosely packed here, but in C4 plant vascular tissues are surrounded by bundle sheath cells, and then most importantly mesophyll cells form a tight packed bundle around the bundle sheath cell. This closely packed circle of mesophyll cell avoid the intrusion of oxygen into the bundle sheath cell. So what happened in C4 plant, when the carbon dioxide enter into mesophyll cell, it is taken up by the phosphoenol pyruvate, which is then converted into oxaloacetate, which is a 4 carbon molecule. That is why it is called as C4 plants, because the first product is being produced here is a 4C molecule oxaloacetate. Then this oxaloacetate is converted into malate, which is transferred into bundle sheath cell, and this malate release the carbon dioxide in bundle sheath, and this carbon dioxide is being fixed by Rubisco through the Calvin cycle, and the starch is being produced. This allows the carbon fixation at mesophyll cell, whereas the Calvin cycle takes place in the bundle sheath. Therefore, it avoids the photorespiration, because if oxygen is entered into it, it will remain in the mesophyll only. It will not go into the Rubisco for bundle sheath cell. Therefore, the H-Slack pathway avoid the photorespiration. And this is how the C4 plant avoid the photorespiration and enhance the photosynthesis. In chem plants, when we see the anatomy, they have stomatas which opens only at night and closes at all the daytime. This opening and closing of stomata avoids the evaporation of water during the daytime, as well as these stomata open at night, so carbon dioxide enters into mesophyll at night and converted into a C4 molecule and store itself in the this molecular form. When the daytime comes, the stomatas are closed and the Kelvin cycle takes place in the daytime. So the oxygen is cannot be entered into mesophyll at the daytime when the Kelvin cycle is taking place here. So chem plants have adapted them to take up the carbon dioxide in the mesophyll only. This is the mesophyll cell only, but the carbon fixation takes place in night, whereas the Kelvin cycle takes place in the daytime in its own mesophyll cells. When we compare the photosynthesis in all three categories, you can see here in C3 plant, carbon dioxide is entered through the stomata. Rubisco has taken up this carbon dioxide and converted into 3 PGA molecule, and this Calvin cycle produce the starch and glucose. Whereas in C4 cycle, carbon dioxide goes into mesophyll, taken up by the phosphoenol pyruvate and converted into C4 molecule. Then the C4 molecule goes into the bundle sheath cell, release the carbon dioxide, and this carbon dioxide enter into Calvin cycle by the adding to Rubisco, and the food is being prepared here in the bundle sheath cell. So carbon fixation takes place in the mesophyll and Calvin cycle takes place in the bundle sheath cell. But chem plants have adapted a different method for avoidance of photorespiration. They open their stomata at night, so carbon dioxide is being taken up by the phosphoenol pyruvate and save as a malate form. But then in the daytime, this malate is used to release the carbon dioxide, and Calvin cycle takes place only at a daytime when the stomatas are closed. Therefore, no oxygen can enter into the mesophyll and therefore avoid the photorespiration. So this is how the process of photosynthesis is different in C3, C4 and chem plants. Here is the basic difference or comparison of the process of photosynthesis. The C3 photosynthesis occurs in the most of the plants. Here are the only 3% of plants fall under the C4 category, and chem plants are mainly cactus and pineapples. And C3 photosynthesis occurs in mesophyll cells only, whereas C4 photosynthesis occur in mesophyll and bundle sheath cell. Chem process takes place in mesophyll only. but the main difference is the first stable product is formed in C3 cycle is 3 phosphoglyceraldehyde, which is a 3 carbon atom molecule, and in C4 and CAM there is first stable product is oxaloacetate. The C3 photosynthesis produces takes up the more NADPH and ATPs, and the C4 and CAM uses most of the energy by the cell. They have C3 plants have typical anatomy, whereas C4 has Kranz anatomy, and CAM plants have xeromorphic anatomy. So this is the basic comparison of photosynthesis in C3, C4 and CAM plants. I hope you have understood the process and anatomy of all these three categories of plants. Thank you very much.